All right, hello everybody, and welcome to our next topic, which is experiments. So we have this broken up into a couple of different lectures. In fact, just two. We have one general experiments and then one on experimental design that we have broken into the different types or considerations in experimental design. So let's start here with just general experiments. So we've talked a little bit about experiments before when we first talked about the three main types of studies in our very first lecture, in our very first topic. There are three main domains of research. We have descriptive research, which is usually observational, or there's no manipulation. Then we have correlational studies, which fall somewhere in the middle. We're looking at the relationship between two variables. But experiments are the last and sort of gold standard of research because what we do is we isolate an independent variable, try and manipulate it in some way, and look at a change on an outcome variable. Remember that our independent variable is a predictor, our dependent variable is an outcome. So if we can't isolate or manipulate our independent variable in a way, we can actually determine cause and effect relationship. So causes are sort of a buzzword here, all right? It's very difficult to determine cause using science and using the scientific method, and this is how we have to do it. We're going to talk about the few facets that make an experiment an experiment. So here, what I had said, we systematically vary the variable that is thought to be causal. That's sort of a funny sentence, right? We vary the variable. But think about it this way, right? I think that a great way to think about this is using drug trials. If we're trying to say, put a certain drug onto market that's going to alleviate symptoms, or let's use something general like depression, right? Our independent variable, the predictor, we're predicting that this drug is going to alleviate or decrease depression symptoms. In order to determine that we have to compare it to something else, we need to systematically vary that variable. That could be like having a placebo group and an active drug group. That could be having a low dose, a medium dose, a high dose, as well as a control group. But that systematic varying is what's really key here. We need to have comparison points to each other. We don't have that in descriptive research. We don't have that in correlational research that's specific here to experiments when we are actively manipulating or systematically varying the independent variable. The idea here is that we hold everything else constant. Any other constant variables, the setting, the environment, we hold as many other things constant as possible so that we can isolate the independent variable, so we can make a claim at the end of the day that the dependent variable was caused by the independent variable. This is, of course, difficult to do, right? We've talked all about how there are so many different variables, subject variables, confounding variables that can impact this, but this is the goal, right? This is the general idea here. All right, so how do we do it? Right, we use random assignment, randomized assignment. So randomized assignment is a little bit different from random sampling. So we'll talk about this in levels here. So let's say let's continue on with this depression example, okay? So we want to take a look at this drug and how it affects those with depression. Well, we could gather a random sample of individuals with depression. That could be like our first level of randomization in the sample. Then we have a sample of those who are struggling with depression. And maybe for this example, let's take the colors, the orange and the blue, to be something like gender, all right, a subject variable that we can't manipulate going into the study. So we would randomly sample, then we would randomly assign those in the sample to one of two groups. The basic experimental design is having a treatment group and a control group. Right? A control group means a group that is not receiving the condition, whatever it is that we're applying to this experiment. They are not receiving it. We've seen some examples in this in the articles that we read for the class, right? So control means that they do not receive whatever the independent variable is, right? They're part of a control group. The treatment group receives the treatment. In this case, if we're using our drug example, they would receive the medication for depression. Then we would do a comparison across these two groups to see if there's a difference. Now, of course, randomized assignments are not perfect. It's not that we have perfectly comparable groups in our treatment and control group always. Due to this, however, if we find a statistically significant difference in depression symptoms between these two groups, we can attribute it. We can say that the drug perhaps caused that difference. Of course, we still need to adhere to the scientific method. We'd want to repeat this process again and again to make sure that this wasn't an error. But this is the general idea, all right? Control all the other variables, use randomized assignment, and manipulate the independent variables. Measure the effect to see if there was an impact on the outcome variable. So this is how we get cause. Remember that cause is a word that's often thrown around in our day-to-day language, right? We say one thing caused another. In science, it's a little more nuanced, right? To say one thing caused another means that we've considered nearly everything else that could contribute to that dependent or outcome variable. So one study is never going to cut it, right? The studies that we do in this class will not determine cause and effect relationships because we're doing something called a quasi-experiment, which we'll talk about in the next topic with experimental design. All right, so here's an example, an image, right? Maybe the experiment is that students' test scores will be affected by distracting sounds in the environment. All right, normally test-taking environments are meant to be quiet and consistent. So the control group would be just a normal class. The experimental group would be the group with distracting sounds, right? So just a visual way to think about this. If we saw a difference, right, we would assume that we randomly assigned students to each of these groups, that we didn't put like the excellent students in the experimental group or vice versa. So if we had randomized assignment, if we compared these and the experimental group did significantly worse, we could suggest, right, we could say that we have some evidence for there being a cause and effect relationship between distracting sounds and test scores. So notice even then when using the word cause, we sort of dance around it. If you remember back to your statistics class, remember we never prove or accept a hypothesis. We just have evidence to suggest it, and that persists here as well. All right, so our simple experimental design, another kind of visualization of this, right? We have two conditions, experimental and control. The experimental group will receive something, the other will not. In this case, we have a mentoring program and outcomes, but I like this particular example because it includes the course of time. So in the experimental group, we see people before a mentoring program, during the mentoring program, then the outcomes as a result of the mentoring program. And then we have another group that follows the same timeline, and we look at outcomes as a result of time, right? We could assume this is an academic mentoring. How much would somebody improve over the course of time naturally, right? We would want that comparison group. So a big issue with, I'll call it pop science. All right, maybe some headlines that you read or snappy articles you find on, BuzzFeed or Facebook or Instagram or Reddit or wherever else on the internet, right? TikTok. Oftentimes studies will be done without a control group. And we're basically looking at a finding that's floating around in space, unanchored to anything. And that's not helpful for us, all right? This happens a lot with, say, the vitamin industry. They'll say, hey, taking this vitamin increases, whatever you're looking at. But they're so baseline, right? It increases from what? It's increasing from what? So we always want to be careful when something's making a claim for a cause or an effect or a very strong claim that they actually have a control group or a baseline to compare to. Again, this is really important in things like pharmaceutical studies because improvement is only impressive if we know what it's improving from, right? We need that control group. All right, so some general types of experimental design here. All right, this is a major distinction we have within subjects design and between subjects design. So let's stick with pharmaceutical examples because they're simple. Sorry, I keep yawning. So let's say that we are going to put this depression medication to trial. So we could do that with a within subjects trial, which is where the same person carries out all conditions. So meaning the control of experimental groups or groups, right? We can have multiple so groups, and we would randomly assign the order of the treatments to the individuals. So for example, let's say that we take each person in our sample that's assigned to the experimental group. We randomly assign which group they're going to go into first. Are they going to be in the control? Are they going to be in the experimental? So, for example, let's take a person who's going to test out this drug, maybe for 6 weeks. We have them take a placebo, and then for the next 6 weeks, we have them take an active ingredient. In this case, within subjects, we're comparing the same person as both a control and as our experimental group. So this eliminates the issue of having to compare across subject variables in a between subjects design, because a between subjects design looks at two different groups of people. One entire group gets the placebo, one entire group gets the active ingredient. So if we think about this a little bit, we can think about the pros and cons of each of these. All right, the pro of a between subjects design, you could have both these groups doing this at the same time, right? You can have 6 weeks of somebody on placebo, 6 weeks of somebody taking the active ingredient. Then you can compare them. If we're going to do within subjects, we would need them to take 6 weeks of placebo and 6 weeks of the experimental design. That'd be a 12-week study as opposed to a 6-week study. So that makes a bit of a difference, right? If you have a time crunch. The argument for within subjects design is that you are the best baseline for yourself, right? Improvement within yourself is the best comparison. It's not always practical. It's not always reasonable. But these are two different designs, right? And we'll talk about these designs and the papers we continue to read in this course. All right, we have something called counterbalancing. Counterbalance has to do with the order in which conditions are given. So I think that this image helps out quite a lot here. So we have a between subjects design on top with two conditions, then within subjects below. So for between subjects, we're just splitting the sample into two groups. One gets the experimental condition, one gets the control condition. And within subjects, if they're receiving both, we want to change the order in which certain individuals are receiving the conditions. So for example, one participant might take 6 weeks of placebo, then 6 weeks of the active drug. Another participant might do the opposite, take the active drug first and the placebo second, in order to eliminate what we call order effects. So with drugs, we could see this, right? Maybe that taking the pill, there would be some kind of crossover into taking the placebo, or even vice versa. But this is even more important when we're doing things like memory tests, the control versus the experimental group. There might be something like a practice effect in place. So counterbalancing all of your participants in this way should help to alleviate statistically the influence of those effects. So I have a little arrow here. This is counterbalancing, is when you switch the order, the conditions. Someone might get the first condition first, the second condition second. The other person might get the second condition first and the first condition second. Okay. All right. Another element of experimental design is this idea of pre and post test. Right? We can do this with multiple types of design. So say that we randomly assign individuals into groups. We give some a pre-test, then we give them the condition, and then we do a post-test. All right, so this is an example of, say, within subjects design. So maybe we're doing an experiment where we're trying to improve our memory. There's lots of gimmicky little games out there that say they'll improve your memory. There are some that are being created by scientists that maybe have some promise. But maybe you're randomly assigned into a group where you're going to be playing a brain training game once a week. So maybe you even start before they give you a pretest. They see where your brain's at, how you're already doing, and then they expose you to the condition, right? You would take part, use the game, the training, then they would do a post-test to see if there was any improvement in yourself. So there are multiple variants. Yeah, multiple variants. All right. I think this helps as well, sort of a flowchart of these things, comparing the experimental group to the control group. So on the left here, we have experimental groups, and then our event, and then the post-test comparison. Control groups won't receive this, right? They won't receive the condition. So this goes back to one of those first images we looked at, looking at the course of time, right? So we compare initially, we see if the experimental and control group are similar. So if we take this brain training example, maybe you take a sample of people and you split them into two groups. You give one group the brain training, one group does not. You could do a pre-test, make sure those groups are the same or similar, right, statistically, insignificant from each other. So similar in terms of, let's say, memory capacity. Then you let time go on. You let the experimental group participate in the training. The control group does not. Then you would compare them at the end. If you found a significant difference at the end, you could say, hey, the game seemed to do something. The training seemed to do something. If not, you can say, hey, both of these groups look the same due to the passage of time. It doesn't seem that the condition that we applied to the experimental group is the cause of any type of change, because there's no difference between the two groups at the end. All right, and lastly, we have matching here. All right, so I think matching deserves to be talked about because it's important, though it seems counterintuitive. So we've spent all of this time talking about randomization, random design, how important it is. But matching is a way of ensuring that the participants in our two groups in an experiment are similar on variables that might be expected to affect the outcome of the study. So let's take some examples. I'll go ahead and use my field as an example, autism research, because we have a lot of these considerations in autism research. So if we have two groups that we want to compare, say we want to compare a typically developing group of individuals to those who are on the spectrum. So we could randomly select from those with autism spectrum disorders, and we could randomly select typically developing individuals, and we can compare those two groups. However, we know that there is some abnormality in the autism spectrum, right? And by abnormality, I mean that it's predominantly men with autism. And then we have another strange consideration when we're doing neuroimaging. I have handedness here. Handedness is important because the brain is lateralized. And if we're going to take a look at the brain, someone who's right-handed will have a brain that's organized differently than somebody who's left-handed. So you don't want to say have 10 left-handed people in your autism spectrum group and none of them in your typically developing group, right? That would kind of offset our comparison. So we have a process called matching, where we basically want no significant differences between our groups on a certain set of variables. Maybe age is another. If we're comparing brain development, we don't want to compare children to adults, right? They'll have totally different brains, so you'd want children in both groups. So matching is something that we can do. Again, it goes against our principle of randomness, our sort of golden standard for wanting random samples. But sometimes it makes our groups more comparable statistically, more comparable, right? We don't want to compare apples and oranges. We want our groups to be similar. Remember, in an experiment, we want to control for almost everything that we can apart from that independent variable. So this can be helpful. Most of us will not be doing any type of matching for our studies, so let that just be known. I mean, if we do it, it might be very limited, right? Maybe you just want equal groups of, say, gender to compare across two groups, which is relatively easy to achieve. Yeah, so that's our general overview of experiments. Our next topic will dive into more specifics of experimental design, so I will see you over there.